Hello friends, I am Dr. Sanjula Babuta from Department of Pharmaceutics, School of Pharmaceutical Education and Research, Jamia Hamdard. Today, we will be discussing about the module Introduction to Biopharmaceutics and Pharmacokinetics. Students, this is a very interesting topic and it is the backbone of the whole biopharm and the pharmacokinetics that you are going to study. In this module, we are going to cover topics related to process of drug usage in disease, biopharmaceutics, concept of bioavailability, concept of biopharmaceutics, biopharmaceutics classification system also known as the BCS, BIOWAVERS, pharmacokinetics, clinical pharmacokinetics, the LADMER model of pharmacokinetics, rates and orders of reactions, and the various pharmacokinetic models. Let us understand the process of drug usage in disease. The pharmacologically active molecule is synthesized, isolated or extracted, and its use rationalized in terms of toxicity and potential benefits. Then its dosage form is made so that a recommended dose can be administered through an appropriate route so that it reaches the site of action or the tissue. Then a dosage regimen has to be selected because of an effective concentration remains in the body, which is influenced by the various physiological, pathological and clinical needs. Dosage form enables the drug to reach its site of action. It delivers the drug at a therapeutic concentration over an appropriate time frame. The study of biopharmaceutics and pharmacokinetics of drug decides whether these goals are being met or not. Now let us understand what is a drug product. FDA defines a drug product as a finished dosage form that contains the active ingredient generally but not necessarily in association with the inactive ingredients. And then what is pharmaceutics? Pharmaceutics is a branch of pharmacy which deals with the study of fabrication of drug into a dosage form. It is a systematic approach to get an effective and stable formulation without disturbing its quality. It deals with technology involved in large-scale manufacturing. Now friends, let us understand about biopharmaceutics. Biopharmaceutics is the study of the interrelationship of the physicochemical properties of the drug, which is also known as active pharmaceutical ingredient API, and the drug product, that is the dosage form in which the drug is fabricated based on the biological performance of the drug. Biopharmaceutics also considers the impact of various manufacturing methods and the technologies on the intended performance of the drug product. It uses quantitative methods and theoretical models to evaluate the effect of the drug substance, the dosage form, and the routes of drug administration on the therapeutic requirements of the drug and the drug product in a physiological environment. One of the major parameters is bioavailability, which is often used as a measure of the biological performance of the drug and is defined as the measure of the rate and extent, that is the amount to which the active ingredient or the active moiety becomes available at the site of action. It is also a measure of the rate and extent of the therapeutically active drug that is systemically absorbed. Biopharmaceutics allows for the rational design of drug products to deliver the drug at a specific rate to the body in order to optimize the therapeutic effect and minimize any adverse effects. It is based on the physicochemical characteristics of the active ingredient, the desired drug product, and the considerations of the anatomy and physiology of the human body. Inherent in the design of a suitable drug product is the knowledge of the pharmacodynamics of the drug, including the desired onset time, duration, and intensity of the clinical response, and the pharmacokinetics of the drug, including absorption, distribution, elimination, and target drug concentration. Thus, biopharmaceutics involves various factors that influence the protection and stability of the drug within the drug product, the rate of drug release from the drug product, the rate of dissolution of the drug at the absorption site, and the availability of the drug at its site of action. The following figures demonstrates the dynamic relationship between the drug and the pharmacological effect. Let us understand the concept of bioavailability. When a drug is given intravenously, it is administered directly into the blood, and therefore we can be sure that the entire drug reaches the systemic circulation. The drug is therefore said to be 100% bioavailable. However, if a drug is given by another route, there is no guarantee that the whole dose will reach the systemic circulation intact. The amount of an administered dose of the drug that does reach the systemic circulation in the unchanged form is known as the bioavailable dose. The percent of an administered dose of a particular drug that reaches the systemic circulation intact is known as the bioavailability. According to FDA's regulation, bioavailability is defined as the rate and extent to which the active ingredient or active moiety is absorbed from a drug product and becomes available at the site of action. Absolute bioavailability compares the bioavailability of the unchanged drug in the systemic circulation following a non-intravenous dose like oral, rectal, transdermal, sublingual, intramuscular, subcutaneous with the bioavailability of the same drug following intravenous administration. The bioavailability exhibited by a drug is thus very important in determining whether a therapeutically effective concentration will be achieved at the site of action. In defining bioavailability in these terms, it is assumed that the administered drug is the therapeutically active form. This definition would not be valid in case of prodrugs whose therapeutic action normally depends on their being converted into a therapeutically active form prior to or on reaching the systemic circulation. It should also be noted that in the context of bioavailability, the term systemic circulation primarily refers to the venous blood and the arterial blood which carries the blood to the tissues. Therefore, for a drug which is administered orally to be 100% bioavailable, the entire dose must move from the dosage form to the systemic circulation. The drug must therefore be completely released from the dosage form, be fully dissolved and stable in the gastrointestinal fluids, pass through the gastrointestinal barrier into the mesenteric circulation without being metabolized, and pass through the liver into the systemic circulation in the unchanged form. Anything which adversely affects either the release of the drug from the dosage form, its dissolution into the gastrointestinal fluids, its permeation through and stability in the gastrointestinal barrier, or its stability in the hepatic portal circulation will consequently influence the bioavailability exhibited by that drug from the dosage form in which it was administered. Many factors have been found to influence the rate and extent of absorption and hence the time course of a drug in the plasma and therefore at its site of action. These include the food eaten by the patient, the effect of the disease state on the drug absorption, the age of the patient, the site of the absorption of the administered drug, the co-administration of other drugs, the physical and the chemical properties of the administered drug, the type of dosage form, the composition and the method of manufacturing of the dosage form, the size of the dose and the frequency of administration. Thus, a given drug may exhibit differences in its bioavailability if it is administered in the same type of dosage form but given by different route of administration. For example, an aqueous solution of a given drug administered by the oral and intramuscular routes by the same routes of administration but different types of dosage forms, like for example a tablet, a hard gelatin capsule and an aqueous suspension administered by the peroral route, or in the same type of dosage form by the same route of administration but with different formulations of the dosage form. For example, different formulations of an aqueous oral suspension shows difference in its bioavailability. Variability in the bioavailability exhibited by a given drug from different formulations of the same type of the dosage form or from different types of the dosage form or by different route of administration can cause the plasma concentration of the drug to be too high, resulting in side effects, or it can be too low, causing the drug to be ineffective. The following figure shows the plasma concentration time curve following a single oral dose of a drug, indicating the parameters associated with a therapeutic effect. The therapeutic window is the drug concentration which are above the minimum effective concentration and below the maximum safe concentration. Poor biopharmaceutical properties may result in poor and variable bioavailability, difficulties in toxicological evaluation, difficulties with bioequivalence of formulations, multiple daily dosing, the requirement for a non-conventional delivery system, long and costly development times, and high cost of the goods. Now let us understand the various terminologies which have been pointed in the figure. The first is minimum effective concentration, which is also known as MEC. This is the minimum concentration of the drug needed at the receptors to produce the desired pharmacological response. The second is the minimum toxic concentration, the drug concentration needed to just produce a toxic effect. Onset time, the time required for the drug to reach the minimum effective concentration is known as the onset time. Duration of action, the difference between the onset time and the time for the drug to decline back to the MEC constitutes the duration of action. The area under the curve, it is related to the amount of the drug which has been absorbed systemically. Cmax, which is also known as the maximum plasma concentration. The maximum drug concentration observed in plasma at a particular time is known as the Cmax. It is usually related to the dose and the rate constants for absorption and elimination of the drug. Tmax is the time at which maximum drug concentration is observed in plasma. It is proportional to the rate of drug absorption. T-half, also known as the half-life of the drug. Administration of a drug with a short half-life requires frequent dosing and often results in patient non-compliance. Half-life is determined by the distribution and elimination. The deferment of half-life can be achieved by increasing the volume of distribution and decreasing the clearance. VD, known as the apparent volume of distribution, it is defined as the volume that would accommodate all the drug in the body if the concentration throughout was the same as in plasma. Thus, it describes the amount of the drug present in the body as a multiple of that contained in a unit volume of plasma. Let us understand the biopharmaceutics classification system known as the BCS. As a result of the plethora and variability of biopharmaceutical properties of the existing and the potential drugs, an attempt has been made to classify the drugs into a small number of categories. A scientific basis or a BCS has been proposed that classifies the drug into 4 classes according to their dose, their aqueous solubility across the gastrointestinal pH range, and their permeability across the gastrointestinal mucosa. The scheme was originally proposed for the identification of immediate release solid oral products or on those on which in vivo bioequivalent tests may not be necessary. According to this, the drugs have been classified into 4 classes according to their dose, their aqueous solubility across the gastrointestinal pH range, and their permeability across the gastrointestinal mucosa. The different parameters which have been included in the BCS classifications include solubility. A drug substance is considered highly soluble when the highest dose strength is soluble in 250 ml or less of water over a pH range of 1 to 8. Dissolution. An immediate release drug product is considered rapidly dissolving when not less than 85% of the labeled amount of the drug substance dissolves within 30 minutes using the USP apparatus 1 at 100 rpm or apparatus 2 at 50 rpm in a volume of 900 ml or less. Permeability. A drug substance is considered highly permeable when the extent of absorption in humans is to be more than 90% of an administered dose based on mass balance determination. According to the BCS classification, the drugs have been divided into 4 classes depending upon the solubility and the permeability. Class 1 drugs are highly soluble and highly permeable. Class 2, drugs having low solubility but high permeability. Class 3, drugs having high solubility but low permeability. And class 4, drugs having low solubility as well as the low permeability. Now let us understand about the class 1 drugs. The class 1 drugs will dissolve rapidly when presented in immediate release dosage forms and are also rapidly transported across the gut wall. Therefore, unless they form insoluble complexes, are unstable in the gastric fluids or undergo pre-systemic clearance, it is expected that such drugs will be rapidly absorbed and thus show good bioavailability. The examples of class 1 drugs are beta blockers like propranolol and metoprolol. Class 2 drugs are the drugs in which the dissolution rate is the rate limiting step in oral absorption. For class 2 drugs, therefore, it is necessary to generate a strong correlation between the in vitro dissolution and the in vivo absorption. The examples of class 2 drugs are the non-steroidal anti-inflammatory drugs like ketoprofen, aceclofenac, and the anti-epileptic drugs like carbamazepine. Class 3 drugs, these are those which dissolve rapidly but are poorly permeable. Examples are H2 antagonists like ranitidine and the beta blockers like atenolol. It is important that dosage form containing class 3 drugs release them rapidly in order to maximize the amount with time these drugs, which are slow to permeate the gastrointestinal epithelium, are in contact with. Class 4 drugs are poorly soluble and poorly permeable drugs. These drugs are liable to have poor bioavailability or the oral absorption may be so slow that they cannot be given by the oral route. The diuretics, hydrochlorothiazide and furosemide are the examples of class 4 drugs. Biowaivers. The biowaiver is a term used in a regulatory drug approval procedure when the application, that is the dossier, is to be approved on the basis of confirmation of equivalence at variance with thorough in vivo equivalence testing. The term biowaiver states that in vivo bioavailability or bioequivalence study may be waived off. Pharmacokinetics. The pharmacokinetics is the study of the time course of ADME, that is absorption, distribution, metabolism, and excretion of the drug. Just as the physical, chemical, and formulation principles are intimately linked with the pharmacokinetic profiles, the pharmacokinetic profile is directly related to the pharmacological activity of a drug. Now let us understand the ADME in detail. The first part in the ADME is absorption. Absorption means the movement of a drug from the site of administration to the bloodstream. The rate and efficiency of absorption depends on the route by which the drug is administered. In most cases, a drug must be absorbed across a biological membrane in order to reach the general circulation and then to elicit a pharmacological response. Even drugs that are dosed intravenously, they need to cross the vascular endothelium to reach the target tissue or distribute into the blood cells. Often, multiple membranes are encountered as a drug transverses through the absorptive layers and diffuses into the bloodstream. Transport across these membranes is a complex process influenced by various things like ionization equilibria, partitioning into and diffusion across a lipophilic membrane and having a potential interaction with the various transporter systems like the influx or the efflux transporters. Membrane transport can occur either passively or actively. Passive transport, known as the diffusion, is the movement of molecules from a region of higher concentration to the one of lower concentration. The membrane permeability, which is directly related to the relative lipophilicity of the drug. It is a major factor affecting the rate and extent of absorption for a given compound and for the gastrointestinal absorption. The concentration gradient is related to the solubility of the compound in the intestinal brush border environment. The concept of permeability, absorption and bioavailability are sometimes used interchangeably, while in fact each represents a different aspect related to membrane transport. Permeability refers to the ability of a compound to cross the membrane. A permeable compound may diffuse across the intestinal epithelium only to be actively transported out of the cell. This compound is permeable yet not absorbed. Likewise, a drug may pass through the intestinal epithelium indicating absorption yet may be metabolized in the gut wall or the liver prior to reaching the peripheral circulation. The drug is absorbed yet it is not bioavailable. Let us now understand about distribution. Distribution is a measure of the relative concentration of a drug in different body tissues as a function of time, and it's related to its ability to diffuse from the bloodstream, tissue perfusion, relative lipophilicity, and tissue plasma protein binding. The apparent volume of distribution, also known as BD, is reflective of the extent of tissue distribution. Drug distribution in vivo is often related to the drug's chemical structure. It can be measured and manipulated during the course of compound optimization by addition or deletion of certain functional group or structural features. However, formulations typically cannot have significant impact on a drug's distribution properties without chemical alterations such as conjugation or use of specific drug targeting technology. The next is metabolism. Metabolism is one of the most important mechanisms that the body has for detoxifying and eliminating drugs and other foreign substances. Drugs delivered by the oral route must pass through the liver before reaching the general circulation. Metabolism at this point is called as the first pass metabolism, which can limit the systemic exposure for drugs despite having good absorption. Oxidation, reduction, hydrolysis and conjugation are the most common metabolic pathways, generally leading to more hydrophilic compounds that can be readily excreted. Cytochrome P450 enzymes are a family of drug metabolizing enzymes that are responsible for the majority of drug metabolism as well as many drug-drug interactions. Although the primary role of metabolism is to facilitate elimination of drugs from the body, secondary effects include transformation of drugs into other active or toxic species, which could be desirable in case of prodrugs or undesirable with respect to toxic metabolites. Elimination of drugs from the body can occur via metabolism, excretion, which can be renal, biliary, respiratory, or a combination of both mechanisms. As with distribution, these phases of drugs PK profile are inherent to the chemical structure of the drug and are optimized during the drug discovery process. Clinical pharmacokinetics. It is the application of pharmacokinetic principles to the patient care which can help the clinician in making rational drug use decisions. However, knowing the relationship between the time course of drug concentration and the pharmacological effect is critical to the application of pharmacokinetic principles and the interpretation of plasma drug concentration in the patient care setting. Clinical pharmacokinetics, on the other hand, is usually limited to a very few and sometimes no plasma drug concentration, requiring the clinician to make an educated guess about the key elements of drug disposition and the drug use process. Let us understand the importance of pharmacokinetics. The studies which have been completed in the laboratory animals may give useful indications for drug research and development. For example, less powerful molecules in vitro can turn out to be more effective in vivo because of their favorable kinetics, that is, greater absorption, better distribution, etc. Pharmacokinetics favors the studies of preclinical toxicology in animals because the drug level in plasma or tissue are often more predictive than the dose to extrapolate the toxicity data to man. Toxicokinetics is also important to verify that the animals have measurable levels of drug in the plasma and that these levels are proportional to the administered dose, estimate the area under the curve and the maximum concentration of the drug in the plasma because these parameters can be used to represent the exposure of the body to the drug, provide evidence about the variability in pharmacokinetics between the various groups of treatment, the days of treatment and other factors, and estimate the variability between animals and identify cases with abnormal levels of the drug. The third importance of pharmacokinetics is the knowledge of pharmacokinetics and of the effects, that is the pharmacodynamics of the drug in man, is necessary for a correct use of drugs in therapy, that is the choice of the best route of administration, choice of the best dose regimen and dose individualization. Now let us understand about the LADMER model of pharmacokinetics. L means liberation, A is absorption, D is distribution, M is metabolism, E is elimination and R is response. Liberation, the release of the drug from the dosage form, that is release of the drug from a suspension, distribution of the drug from emulsion droplets into the body fluids and diffusion of the drug from an implant. Liberation is usually assessed in vitro with the help of dissolution test. Liberation is automatically guaranteed when the drug is administered as an aqueous solution. Absorption. The key elements include the site of absorption, its mechanism, and the factors that influence absorption. In case of IV administration, there is an immediate onset of action and the liberation and absorption steps are essentially instantaneous. Distribution. It is used to describe the process by which a drug is reversibly transported from one tissue fluid to another within the body. Distribution can be within the blood, distribution can be between the blood and the other tissues. Next is metabolism, also known as biotransformation, where the drug is converted from the active form to the inactive form in majority of the cases. The last step is the elimination of the drug, which is through the kidneys known as the renal elimination, through the liver known as the biliary elimination, intestinal elimination and the pulmonary elimination. Pharmacokinetic models. Nowadays, there are new sensitive, accurate and precise analytical models for the direct measurement of the drug in biological sample like plasma and urine. The pharmacokinetic is able to use these measurements of drug concentrations to describe more accurately the bioavailability differences among the drugs and the drug products. In order to achieve therapeutic response, plasma concentration of drug should be maintained within the therapeutic window. For this, the data obtained by analysis of biological fluids is used to study the kinetics of the processes involved for the drug absorption, distribution, metabolism and excretion of drugs. The various pharmacokinetic models which have been used are the compartmental models, the physiological models and the non-compartmental models. The primary objective of pharmacokinetic models is to develop a quantitative method to describe the relationship of drug concentration or amount in the body as a function of time. The complexity of the pharmacokinetic model will vary with the route of administration, the extent and duration of distribution into various fluids and tissues, the process of elimination and intended application of the pharmacokinetic model. There are a wide variety of potential uses for pharmacokinetic models, which includes prediction of drug concentration in blood plasma or tissue, calculation of a dosage regimen, quantitative assessment of the effect of the disease on drug disposition, elucidation of the mechanism of disease-induced alteration in drug disposition, determination of the mechanism for drug-drug interactions, and prediction of drug concentration versus effect relationship. Let us understand about these various pharmacokinetic models. The first are the compartmental models. A body can be represented as a series or systems of compartments that communicate reversibly with each other. A compartment is not a real physiologic or anatomic region but considered as a tissue or group of tissues which have similar blood flow and drug affinity. Within each compartment, drug is considered to be uniformly distributed. Mixing of the drug within a compartment is rapid and homogeneous so that the drug concentration represents an average concentration and each drug molecule has an equal probability of leaving the compartment. Compartment models are based on assumptions using linear differential equations. The model is an open system since drug can be eliminated from the system. It is assumed that the rate of drug movement between compartments, that is the entry and the exit, follows the first order kinetics. The second are the physiologic models. Physiological models, also known as blood flow or perfusion models, are pharmacokinetic models based on known anatomic and physiological data. Physiological models attempt to describe drug disposition in terms of realistic physiological parameters such as blood flow and tissue partition coefficients. The real significance of the physiological based model is the potential application of this model in the prediction of human pharmacokinetics from animal data. The mass of various body organs or the tissues, the extent of protein binding, the drug metabolizing capacity, and the blood flow in the man and other species are often known or can be determined. Thus, physiologic and anatomic parameters can be used to predict the effect of drugs on man from the effect on animals in cases where human experimentation is difficult or restricted. The third models are the non-compartmental models. They are also known as model independent methods and does not require assumption of specific compartmental model. This model can however be applied to any compartmental model provided the drug or the metabolites follow linear kinetics. The approach are based on statistical moments theory. This involves collection of experimental data following a single dose of drugs. Non-compartment model describes the pharmacokinetics of drug disposition using time and concentration averaged parameters. Now let us summarize this module. Under this module, we have discussed about the different terminologies used in biopharmaceutics as well in pharmacokinetics such as MEC, minimum effective concentration, MTC, minimum toxic concentration, Cmax, Tmax, AUC, etc. According to FDA's regulation, bioavailability is defined as the rate and extent to which the active ingredient or the active moiety is absorbed from a drug product and becomes available at the site of action. A thorough understanding of the biopharmaceutic properties of a drug candidate is important both in the drug discovery setting, where potential drug candidates are being considered, and in the developmental setting, where it is important to anticipate formulation and manufacturing problems. The influence of variability and bioequivalence issues on clinical results must be studied to provide assurance to the regulatory authorities as to the robustness and quality of the drug substance and the drug product. The pharmacokinetics is the study of the time course of ADME, that is absorption, distribution, metabolism and excretion of the drug, whereas clinical pharmacokinetics is the application of the pharmacokinetic principles to patient care, which can help the clinician in making rational drug use decisions. Nowadays, there are new, sensitive, accurate and precise analytical models for the direct measurement of the drug in the biological samples like plasma and urine. There are three primary types of pharmacokinetic models, such as the compartmental, the non-compartmental and the physiological models. Thank you.